So yes, that's right. I'm Dan Ward, and this is a talk about Tautog, a species that we've been looking into for alternative production. So as it was mentioned, I have a company, Ward Aquafarms. We produce commercially shellfish from throughout Cape Cod. We're a vertically integrated company, which means we do hatchery work, we do grow out, we do sales, we sell seed, oyster seed, bay scallop seed, quahog seed to different producers throughout the region, as well as municipalities throughout the region. And yes, we do a lot of work with different research organizations in terms of being able to benefit the U.S. aquaculture industry. And so the rationale between the work that we're doing today is that I work with the commercial aquaculture industry throughout Massachusetts and try to evaluate problems that they have and ways that we can work together, whether it's with us or with another research group, to kind of find those solutions to those problems. So sometimes that's impacts of harmful algae blooms, sometimes that's different hatchery techniques. In this case, it's the fact that as an industry, at least on the East Coast, most of what we produce is oysters for the most part, shellfish in generally speaking. And in Massachusetts, we used to be a little more diverse, but as oyster aquaculture has gotten easier to do, 96% of what we produce in Massachusetts is oysters, right? So an over-reliance on one particular crop, no matter where you're farming, is not a good thing. And we certainly learned this through COVID, that relying on one crop is not good. It's not sustainable for the industry, and it's not sustainable for any of our businesses. So we've diversified into hatchery production and bay scallops, and we work with many farmers throughout the region to get them to grow bay scallops and teach them how to do it and provide them with the resources to be able to do that. This is another example of an opportunity for us to diversify what we grow in Massachusetts. Not necessarily us in particular, but to provide the background to be able to say, is this a good candidate species? So Tautog is a marine wrasse. It's native to Massachusetts and the East Coast down through Delaware. Commercially, it's a valuable species. It commands a high value in the fish market, but also it's even more important in the recreational fishery. It's a very popular food fish. It's highly well received in the market, and however, it's either, depending on when you're looking, it's either overfished right now or it's at least has high pressure to overfishing because there's high site fidelity. So that high site fidelity causes it to have that kind of impact on pressure, right? And so because it's well established that it has a demand in the market, there's overfishing occurring, or at least pressure occurring, and it's very hardy. It's very well known in the Northeast. That's kind of what we look for as a candidate for aquaculture, right? People want it, but they can't get it. So if we can grow it, it's a good candidate that we could potentially do. So what we did first is we worked with University of Massachusetts to do studies on collecting wild Tautog at a smaller size and then growing them up, right? So the first kind of step of this, because there hasn't been any work on Tautog out there. In the early 2000s, there was a couple of projects that happened at a NOAA lab in Milford, but those people have moved on to other roles, and so we couldn't really learn much from them, and there really is nothing else out there in terms of Tautog aquaculture. So we were kind of starting from scratch here. So the first step was saying, all right, well, what do they look like in captivity? What kind of growth rates do we get? How well do they survive? So we started with wild capturing young year one animals, actually directly near some of our aquaculture sites. They were around 5 to 10 centimeters. We graded those out, get a population with a much tighter range than that, and then did grow out studies with them. And so this particular project was kind of cool because it was a mix between, let's see if this is a good candidate species for aquaculture, as well as green crabs are a big problem in the Northeast. So what's really cool about Tautog is that their preferred food source is green crabs. So in the fishery, that's actually what they use as bait for rod and reel fishing is green crabs. So if green crabs are a big problem and are causing issues with wild shellfish mortality, as well as aquaculture shellfish mortality, as well as many other problems that I'm sure most of you know, if we can find a use for green crab as well as growing Tautog, it's a win-win, right? And so there's a lot of details associated with this, but the long story is we fed a green crab or a short story, is we fed a green crab diet, a pellet diet, a just regular commercial shredding diet, and then a mix of two where we went back and forth and did a 50-50 mix. The green crab was by far the best, right? And so what we were able to determine is, while in the wild these fish are known for a slow growth rate, that's primarily due to the site fidelity. And so because of the site fidelity in Massachusetts and through Delaware, it's very cold for a large period of the year. So if they're not really growing for a large period of the year, that contributes to the slow growth rate, which was our hypothesis going into this, is that because there's some evidence that there are certain wild populations that have a much faster growth rate. So our hypothesis was this is a site fidelity thing and not necessarily a genetically slow growing species. And so what we found is by month 5, we were getting a gram a day of growth out of these fish, which is fantastic. So we went from a 5 to 10 centimeter fish to a 1 pound fish that is desired, that's the size desired in the live market, which these are very highly desired in the live market as well, in 9 months. So it's pretty impressive. So if we can figure out all the other pieces associated with this, this is a good candidate for aquaculture. It's very hardy, isn't prone to disease, is able to be handled very well, and has a decent growth rate. So what we found is the crab diet was the best. So we were feeding a ton of crab to these guys, and they were growing really well, and they loved it. Voracious feeders, going crazy for it. The downside is it wasn't good for us humans. It was really ruining water quality. So feeding that much green crab, we would quarter it, freeze it, and feed it out to satiation, led to some pretty extreme water quality issues. So what we learned from this is, great, it is possible if we can figure out a way to feed them that's not green crab, but also green crab is not the solution. So while we love to have a double win of this is a good candidate species and let's solve the green crab crisis, we're not solving the green crab crisis. It really was bad for water quality. So also another thing that led to is some size differentiation and hierarchies developing because there was no structure in the tanks. And this is a wrasse species that lives within cobble and eelgrass naturally. So we started to think about why are these hierarchies developing and what can we do about them. So the next project that we did with the UMass is, all right, well, if we can't feed them green crab, but they like green crab and they don't seem to be feeding on the pellets as well, what if we, because pellets have menhaden in it and they're not usually feeding on menhaden, the menhaden kind of flavor from the fish oil, what if we did a feed attractant trial? So we did a feed attractant trial where we did pelleted diets coated with a squid hydrolysate feed attractant, a green crab hydrolysate feed attractant, and a control. We didn't see a huge difference between them, in between those different diets, but again, high variability throughout. And so the high variability is at least partially due to us not grading them. So I can tell you as a shellfish farmer, if I have a bag of oysters and I don't grade out the large ones, what you end up with is a handful of large ones and 100 small ones. And that's not due to a hierarchy. That's just due to consumption of food within the bag, right? So yes, potentially what we're looking at is hierarchies here, but it also simply could be a reflection of us being researchers and doing this as a research level project and only having so many animals and not grading. Whereas if we treated this as a commercial farm, and any commercial fish farm grades their fish all the time to be able to have different cohorts. So there's a great chance that this hierarchy issue and the issue of size differentiation could simply be solved by actually using standard commercial procedures. The next work we did is some work that we've done for the last 2 years with a NOAA lab in New Jersey at the NOAA Northeast Fisheries Science Center in Sandy Hook. And so this work is saying, all right, well, if we can take them from juvenile 1 year size up to grow out in 9 months, that seems pretty good as a candidate aquaculture species. What can we do the other half, right? Because collecting these things in the wild is not a sustainable way to actually have a candidate aquaculture species. We have to be able to close that life cycle all the way from broodstock through actual sale. So now we kind of started with the second half, now we're moving back to the first half. And so what we did is worked with commercial fishermen in Woods Hole to fish trap us broodstock, high quality fish. Because they were fish trapped, there was no damage to them, very high quality. We moved them to a holding pen for a week or so to make sure that they were all right from a health perspective, and then transferred them down to the lab in New Jersey. Held them at ambient over winter, fed them a mix of capelin squid and a commercial broodstock diet, and then let them condition naturally as water temperature increased. conditioned them naturally, and then did spawns in the spring. So we did volitional spawning as well as strip spawning, and was able to grow those out. Rotifers Artemia pellet, your kind of traditional conversion through that. We had great success through that. We were able to do 2 different year classes of this with improvements in year classes in year 2 as compared to year 1. Raised them at 19C, 25 parts per thousand. Again, not all of them took to the pellets, but a lot of them did. Enough of them did for us to be able to do these juvenile experiments moving forward. There's more work to be able to be done in terms of mortalities during that first 48 hours. But they grew quite well, they grew quite fast, and at 110 days post-hatch, 24 millimeter total length, which again is pretty good compared to your other kind of commercial candidate aquaculture species. The subsequent work that we did is on utilizing different structures to be able to look at hierarchies and kind of size differentiation. When we looked at this, what we actually found is that the control mechanism of no structure actually led to the same survival and growth as either having the rigid structure of these PVC tubes to mimic cobble, or the soft structure of fake eelgrass. And then in subsequent experiments, we actually found that over a 12-week trial using those same structures, that the no shelter ended up being the best in terms of growth too. So what's nice about this is that this leads us to be able to say we don't need to have structure in there. The hierarchies that are developing are probably due to grading and not using standard commercial techniques as opposed to research techniques. We didn't have any disease problems through this. They're able to be handled. They grow fast. They get to market size within 2 years. So it's a great candidate. So to be able to continue this work, hopefully other people will take it on. We'll continue to take it on and continue to look at this because it's well received in the market, and it can be grown all the way through broodstock through final grow out. And I just want to say thanks to my collaborators and my funders through Northeast SARE and NRAC. And yeah, thank you very much. I'm sure there was a lot. There was a lot there. I can answer questions after too. Yeah.
But there's been a lot of work with black sea bass, and obviously we prefer for a lot of reasons strip spawning.
Mm-hmm.
Strip spawning provides a lot lower quality. Did you see the same type of stuff with traditional and strip spawning and egg quality, and then hatching success, fertilization success, and so forth?
So the volitional spawning led to higher survival than the strip spawning, but a lot lower egg volume. So what we were doing is the volitional spawning early, and so that was more of a May thing. And during May, it was going, but then it started to look into, well, are we going to get enough animals out of this to be able to do the subsequent experiments? And so we had to switch in early June to the strip spawning. And so by doing that, yeah, we got a lot more egg volume, lower survival throughout 48 hours, but because of the kind of higher egg volume overcame the limitations of the lower survival. So it was worth it. Commercially, would you do it that way? Remains to be seen, but it worked for our needs for this experiment.